So that means that the endospore is dormant anyway. So until the endospore becomes reactivated, you're not going to see the result of the UV light. But UV light exposure would have destabilized that DNA, would have mutated the DNA, so that when the endospore, or if the endospore comes back as a vegetative cell, the vegetative cell will not be able to reproduce. It will be mutated. Yeah. And then the results of the experiment should give you death of the exposed colonies. I guess it depends on what time, the time length that you use, and as to what the results are going to be. But even if you did not choose a long enough time to show complete destruction, you should probably have seen a reduction in the number of colonies. And it may take as long as 24-hour exposure to completely destroy some organisms. So the way that UV light is used to sterilize is it's used to sterilize rooms, for example, like operating rooms. So when there's nobody in the room, they turn on UV lights, and that sterilizes surfaces. And so that's going to be maybe a 20-hour exposure, 12-hour exposure, however long that room is empty. We also use it in labs. The hoods under which you work, when you're working with bacteria, you lower the door of the hood and you turn on a UV light. The UV light will sterilize the surface. 12 hours later, you come back to work, turn off the UV light, that surface is sterile. Okay, so you should be able to explain the results obtained. And was UV light capable of effectively destroying the organisms tested? Well, that depends on your results. So maybe if you only chose for time, maybe 5 minutes, 10 minutes, you may or may not see complete destruction. If you chose 10 minutes, 20 minutes. Really, UV light doesn't really sterilize water. It disinfects water, but it doesn't sterilize it. It will sterilize surfaces. Yeah, because of diffusion, it still will not penetrate enough to sterilize. It will definitely, and it's used as a disinfectant of water, but not as a sterilant of water. So yeah, that's a distinction there. So yeah, so there are several graphs that you could use. You should include a picture of the graph. Okay, I don't have that as the, I guess a bar graph would probably have been one of the best for this kind of data. And again, the results are going to depend on the organisms that you had and the amount of exposure. Okay, so let's see now, chemical control. Yeah, zone of inhibition, UV light resistance correlate with heat resistance. No, it doesn't. No, it doesn't. Yeah. Obviously, endospores will be heat resistant also. They're UV light resistant, but there is no real correlation. The way heat is going to work is going to affect membranes. It will eventually obviously affect DNA, but not as targeted as UV light does. UV light is very targeted, affecting DNA only. Heat is going to begin melting the membranes. Zone of inhibition will be the zone of no growth of no growth bacteria around the chemical. And then the lawn of bacteria refer to how we inoculate the plate, covering every inch of the plate with the organism. So we inoculate every single inch of the plate with the microorganism, so that when the bacteria begins to grow, it actually grows absolutely everywhere. So we put the discs with the chemical, and we looked at the areas where there is no growth, where the bacteria stop growing, and we measure the zone of inhibition around that area. So let's say that one has nothing, and let's make this 2 nothing. So at least the chemicals, only bacteria can be cultured, right? Yes, correct. Chemical used to paper disc. Okay, so we try chemicals uses disinfect. Oh, okay, so soap, especially if it is antibacterial soap, I guess that would be an antiseptic. Honestly, if it doesn't have any form of antiseptic in it, then it's just a soap. It's really just a degerming, but that's okay. I think I think the answer there that they're looking for will be antiseptic. Iodine is an antiseptic. It could also be used as a disinfectant, but we typically use it as an antiseptic. Bleach is definitely only a disinfectant. Water is the control. Mouthwash is an antiseptic. Hydrogen peroxide could be both, an antiseptic and a disinfectant. The 2 organisms given for the experiment, and I believe in this case, pretty much everybody had the same organisms. Oh, it says no. I have in my notes that organisms will vary. So I guess not everybody has the same organisms. Okay, that's fine. Gram reaction shape, you're going to look that up. And again, that will depend on the organism. Temperature of incubation, it will be at 37 degrees. That's a standard. All the organisms we're working with are medically important, and they're mesophiles. Time of incubation, at least 18 hours, 18, 24, 48 hours, but at least 18 hours. That's the minimum. So here the idea is to get the diameter of the zone of inhibition. So first of all, a couple of things. We're measuring the diameter of the zone of inhibition in millimeters. So that's what I'm going to be looking for. That you did use millimeters, and that it was measured from one end of the zone of inhibition to the other end of the zone of inhibition. So this is the disc, and the zone of inhibition is the circle right there. The diameter of the zone of inhibition is going to be measured from one end across, including the disc, to the other end. The disc itself is about 6 millimeters. So if you tell me that a zone of inhibition is 5 millimeters, well, that's wrong, because the disc by itself is about 6. So there is no zone of inhibition if you're giving me 5 at the zone of inhibition. So typically what that could be is that you're not meaning millimeters, perhaps you measure in centimeters, in which case that would be 50, not 5. So I will be looking for that, the correct units, and that the zone of inhibition makes sense. Which chemical is the most effective against each of the microorganisms tested? The most effective chemical will be the one that gives the biggest zone of inhibition. So let me go ahead and have a little bit of an example. I took pictures. This were smaller in the lab. Okay, well, that's partly why I asked for pictures. That way I can take a look at it and make sure that the answers make sense. Okay. An actual disc is about 5 millimeters. So maybe they were a little bit less, maybe 2 or 3 millimeters, because these were pictures of discs. So let's see. So we have results. We want to interpret the results. So let's say with water, there was no zone of inhibition. Oh, never mind, that's not water, that's hydrogen peroxide, sorry. Okay, so hydrogen peroxide, let's say the zone of inhibition here was 25 millimeters, and let's say here it was 15 millimeters. Water, let's put 0 on that one. Bleach, let's say here it was 30 millimeters, and here it was 37. No, wait a minute, I'm trying to do something specific here. Let me do these. Let me go ahead and change this number. And let's make this one 15. Yeah. So let's make this one 15 millimeters. Let's make this one 10 millimeters. 137. Mouthwash, let's say 9 millimeters, and let's say 13 millimeters. Iodine, give me a second. And then let's say, I don't know, let's say 18. And here, let's say 12. And soap, let's say 10, and let's say 9. Okay. All right, so smethosol, which is most effective against each of the microorganisms tested? Which chemical is most effective against each of the micro? Okay, so a chemical that is the most effective against each of them. Well, most effective for this is organism A, this is organism A. For organism A would be the hydrogen peroxide. For organism B would be bleach. Okay, which is most effective against both? Okay, so if you want to kill A, you want to use hydrogen peroxide. If you want to kill B, you want to use bleach. However, what if you want to kill both? Then you want a chemical that is effective against both. Okay, so which one is effective against both? You could use hydrogen peroxide because that's very effective against A and pretty effective against B. Bleach will be very effective against B, but not as effective against A, so that might not be a good choice. Iodine will be pretty good too. It's pretty effective against both. Okay, so I would say in this example, either hydrogen peroxide or iodine would be good choices. The least effective of each organism overall, not counting water, I guess the least effective would have been soap. Yeah, soap would have been the least effective because you have very low numbers on both. The more sensitive of the organisms is to a chemical. Oh yeah, the more sensitive the organism is to a chemical, the smaller the zone of inhibition. So which means that essentially you would need a, say that's this zone of inhibition versus this zone of inhibition means that you need a higher concentration of the chemical to kill the bacteria. So in this example, because you have a smaller zone of inhibition, the place where the zone of inhibition begins represents a higher concentration of the chemical than in this case on this chemical. Let's say chemical 1 versus chemical 2. Chemical 2, the zone of inhibition is much larger, so that the chemical diffused more further away from the disk, and the concentration at which the organism is killed is a lower concentration for chemical 2 than for chemical 1. All right, so the idea is you have a chemical in the disk, and the chemical begins to diffuse away from the disk. The further the chemical moves from the disk, the less concentrated it is. So if you look at chemical 2 over here, it is less concentrated than chemical 2 over here is more concentrated because it's closer to the disk, which is the highest concentration. The further from the disk, the less concentrated the chemical is. So the question is, at which concentration do you begin to kill the bacteria? That's what the zone of inhibition is giving you. So the zone of inhibition gives you the minimal concentration at which the organism begins to be killed, because there is no growth after that concentration. So this is the minimal concentration right here. Let's say concentration, I don't know, Z is the minimal concentration at which the organism is killed by the chemical. If you give it more after this concentration, there is growth. So let's make growth as something purple. Okay, so this is the bacterial growing, and the bacterial stops growing when the chemical is at a minimum concentration that kills it. So in this case, the concentration that kills it is lower than for chemical 1. For chemical 1, the bacteria get much closer to the chemical, Therefore, you require a higher concentration of the chemical to kill the bacteria. So the organism is more sensitive to chemical 2 than to chemical 1. Because a lower concentration of chemical 2 kills it, you need a higher concentration of chemical 1, the bacteria is more resistant to chemical 1. So what that means is that the more sensitive the organism is to a chemical, the larger the zone of inhibition, which essentially means that a lower concentration of that chemical kills the bacteria, because the bacteria is very sensitive to the chemical. Does that make sense? The larger the zone of inhibition, the more sensitive the organism. Correct. Did I do that just now, or did I do it on Monday whenever I was explaining this lab? Oh, just now. Oh, I'm sorry. Oh, I see what you're saying. Right there, you're absolutely right. I'm so sorry. Oh, darn, did not mean to do that. Oh, I was gonna put a, oh, I did not mean to do that. Okay, all right. The more sensitive the organism is to a chemical, the larger. Yeah, I see what you're saying. No, I'm so sorry. I'm so sorry. Yeah, no. No, I'm glad I continue explaining it. Thank you. No, the larger the zone of inhibition. Yes, hopefully that is what makes sense, because that's what it is. Okay, so the larger the zone of inhibition, the more sensitive the organism, because that means, yeah, thank you. Okay, thank you for pointing that out. Okay, I had not realized I said it wrong the first time around, and you're right, I even circled the wrong thing. I was probably thinking concentration and not zone of inhibition. Yeah, very good. Okay, so the same principle applies for these Kirby-Bauer tests. The only difference here is that we're using antibiotics and not disinfectants or antiseptics. And reaction. Okay, yeah, same thing here. Just look at the organism, and we do need to interpret the zone of inhibition against the manufacturer's chart of resistance, intermediate, or sensitivity. So which of my organisms test is more resistant? And let me see if I have anything on my notes about that, because again, I think everybody had a different organism. But in general, let me see what I have. All right, give me just a second. Yeah, answers might vary. Okay, and I have not done this exercise since last semester, since the summer. So I don't remember what were the possible microorganisms. Having said that, there are some microorganisms that are definitely more resistant than others. So, for example, Pseudomonas aeruginosa, it is extremely resistant. So if anybody got that organism, I would expect that the organism is very, very resistant, most of the antibiotics, except for maybe 1 or 2. Staph aureus, let me see, what are the E. coli and Pseudomonas. Okay, all right. Agalactia, that's a gram-positive. Usually it was P. agalactia, it was not S. Thank you, S. agalactia. Yeah. Okay, that makes sense. It's strep. There you go. Yeah. Okay, so essentially you had a gram-positive. Your group B is strep. Yeah, so you essentially had a gram-positive and a gram-negative. And so let me go for the antibiotics. Yeah. So you have that. What that means is that some antibiotics would not have been effective against the gram-negative. So vancomycin, for example, is a narrow spectrum antibiotic. It should not have been effective against the E. coli. However, vancomycin, if you have Staph aureus, Yeah. So for example, if you had a Staph aureus as your organism, some Staph aureus may be resistant to vancomycin, but I don't know if the exercise gave you a resistant vancomycin Staph aureus or not. Let me go back to my notes here real quick. What else do they have? Another one, let's see what other, there we go. Okay, erythromycin is another one that's narrow spectrum. It's typically used for gram-positives. Obviously, penicillin is narrow spectrum, no question about that one. And as a matter of fact, penicillin, most organisms are even it's a narrow spectrum gram-positives, but even among the gram-positives, most gram-positives are resistant, except Streptococcus. Streptococcus is still very, very sensitive to penicillin. Tetracycline is broader spectrum. Sulfa drugs are broader spectrum. However, with tetracycline, you should have used the correct organism to read. So with tetracycline, we have gram-negative enterics. That would be your E. coli. Pseudomonas has actually that doesn't have a different reading. Gram-negative and Pseudomonas have the same reading. Staph aureus and Streptococcus have a different reading for the antibiotics. So I would have to make sure that you chose the correct value to read the zone of inhibition. Again, sulfas are across the board. So we were dealing with erythromycin, penicillin, and vancomycin as narrow spectrum drugs, and then gentamicin, tetracycline, and sulfa drugs as broader spectrum drugs. Okay, so essentially what I'm going to look for in there is make sure that you read the zone of inhibition correctly and you interpret the result correctly. Yeah, the narrow spectrums were erythromycin, vancomycin, and penicillin. Again, determine you're welcome. Determine the best and worst antibiotic for use. That will depend on your results. But obviously, you would choose an antibiotic for which the organism is sensitive. If there's more than one that could be chosen, then you can put either or both. Doesn't matter. If there isn't any, if any of you maybe had a Pseudomonas, for example, that was resistant to everything, then you don't have a choice. There's nothing to choose. If you have an organism that is resistant, but there is an intermediate, then intermediate is not. So let's say you just have resistant, 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 and one is intermediate. Well, intermediate is not ideal, but since you don't have anything, then you would have to choose the intermediate. I think everybody should have had a sensitive. Okay, so let's see what this one is. Oh, okay. The organism test to determine if there is anything wrong with the results and explain a possible reason for the results. Okay, so let's see the results for organism B, Bacillus nectarium. Okay, so this right here would be your, more than 20. Oh, okay. Number of exposed, number of colonies in control. Okay, so this is your control. So obviously the control, that's good. So the control is what you're going to be comparing against. Plate 2 was exposed to 1 minute and there's no difference there, okay? Plate 3, 20 minutes. Now we definitely see a difference, so there's no growth. So the minimum exposure as far as the experiment, the minimal exposure for this organism to see no growth is 20 minutes. However, when you look down here, 45 minutes, all of a sudden you have less than 20, which is obviously, there's something wrong. They couldn't just kill it at 30 minutes and then at 45, all of a sudden it survives. All right, so a couple of things. What could have gone wrong? It could have been that the plate might have been mislabeled. Let's say that these two were mislabeled right there, and they were switched, in which case, really, when you see something like these, you can almost argue the whole experiment is invalid because you don't know what happened. Unless you can figure out what happened, the whole experiment is invalid. You may have mislabeled these two plates right there. That could be one. Another possibility that this is a contaminant, for example. So what is growing is not your organism, it's something else. Another possibility could be that it was not exposed for 45 minutes. Maybe there was something wrong with the timing. So that's just three things of that I can think of that could have gone wrong there. The other one, again, one that's the control. At 1 minute, the same thing, it still lived. At 20 minutes, it's still, oh, wait a minute, take a look at these. You have now no growth on the control and more than 20 colonies on the exposed. That makes no sense. The control all of a sudden is invalid. So what could have gone wrong? Maybe they were mislabeled. And this one was actually the control, and the other one is the one that was exposed. That could be a possibility right there. Now there is no growth at 30 minutes. You don't have a valid result in 20, so you cannot really say that 30 is the minimum exposure. Really, what this tells you is the whole thing is wrong. Now, if you do say 30 minutes is the minimum for exposure, I probably will take that answer. As long as you explain what happened here, that this, as long as you catch the fact that this is definitely a wrong result. Okay, so here, how you can interpret, you need to explain what went wrong, no, you could need to catch the fact that something went wrong, and then maybe explain what could have gone wrong, what could be one possible explanation. Now, if you say, okay, this is the first one that I see a result that I can trust, and there's no growth here, so at least 30 minutes for sure will kill the organism, whereas the other one, it is 20 minutes. You could say, okay, this organism is more resistant to UV light, appears to be more resistant to UV light than Bacillus megatherium, which was killed at 20 minutes. So you could interpret that if you want. Main thing I'm looking for in here is that you catch the mistakes and you explain possible reasons for the results obtained. That's really what I'm looking for. Okay. All right. Any questions about these? Okay. Let me go then and start with chapter. Oh, you're very welcome. Start with the lab report 8. I'm trying to see which one would be the best. Just a second. So I have 2. Should have more than that. Okay, give me a second. Should have more than these. Give me just a second. I think there's another. So the lab report 8 and lab reports 9 and 10 are going to deal with genetics essentially and DNA. Give me a second. That there. In biology and technology. Okay, I have that one. DNA isolation. Okay. And oh, there we go. All right. So the first thing here is to go back to the structure of DNA. Which we have seen already in chapter 7. I just want to see, this is just, okay, so this is basically going off of chapter 7, really. You should review what is DNA replication, what is transcription, what is translation. Okay, we got all these. All right. DNA isolation. Okay, there we go. That's a good one. All right, so the first experiment you do here is DNA isolation from strawberry cells. And so the first thing, if you want to isolate DNA, is to rupture the cells. And they are the way that this is done is by mechanically disrupting the cells, crushing the cells, and then the cells are mixed with a solution of detergent and salt that further is going to disrupt the cell membrane, and then allow for the inside of the cell to come out. So does water count as a reagent? If it is part of what you're putting in there, yes, it would. So that, see, so you're going to have, and typically they would say DI water, for example, as part of what you need to put in there. So DI water, the detergent, and the sodium chloride. So what's that being in the bag is going to be the cell components and the DNA. So the next step will be to separate the DNA from the cell components. Okay, so let's see. Yeah, we did all these. We disrupt, because we're dealing with strawberry DNA. It's a plant, and plant cells have a cell wall. So we're going to destroy the cell wall, the cell membrane, and it is a eukaryotic cell. So we're going to have to go through we're going to destroy the nuclear envelope to be able to get into the DNA. That's where the detergent comes in, because you're disrupting a lot of membranes, and that's what the detergents do. Keep in mind, membranes are lipids. So the cell membrane and the nuclear envelope are lipids. Detergents emulsify lipids. They destroy lipids. Okay. So that's one thing. The cell wall is cellulose and protein. Let's see. So we got that already. Things are okay. So we're going to filter. And the filter is going to trap all the larger components. Smaller molecules is what is going to be collected in the filtrate. Now we need to separate the DNA from the other smaller components that have been filtered with the DNA in the filtrate. We're going to use detergent, salt, and water. The salt is going to neutralize the negative charges of DNA. Now, when you go back to the molecule, to the DNA molecule, the negative charges are part of the phosphate oxygen groups. That's where the negative charges are located. So DNA is negatively charged. So in order to, salt is sodium chloride. So salts have a sodium, no, it will be positive, negative. So salt is sodium chloride, which means that it has a positive and a negative ion. Keep in mind, in water, this is not, I don't know why it's not letting me write. In water, salt is going to dissociate. So salt is going to dissociate into sodium, and chlorine. Chlorine is negatively charged. So chlorine is negatively charged. Salt is positively charged. These two are going to dissociate. So the positively charged sodium is going to cover DNA or be attracted to the negative charges of DNA. So DNA will be neutralized by the sodium. Again, the detergent is going to emulsify the lipids. The salt is going to separate the DNA from everything else by precipitating it. Keep in mind that anything that has a charge can be dissolved in water. If we want to precipitate, we want to hide the charges, so that precipitation means separation from water, so that we can separate it from the water. So that's the purpose of the salt, is to separate the DNA from the water. Otherwise, it will continue to be. Yeah. Something that dissolves in water is the molecules that dissolve in water are charged molecules. Anything that has a charge is hydrophilic. It likes water and dissolves in water. If you want to precipitate something, you have to eliminate the charges. So because precipitation means separation from water. And the only way to separate from water is to cover the charges. Because water is a charged molecule and it likes other charged molecules. So water has a positive and a negative. We have 2 hydrogens and 1 oxygen. The oxygen is negatively charged. The hydrogens are positively charged. So they are going to attract charged molecules. And that's what dissolves things in water, is that attraction. If you cover the charges of a molecule, now you're going to separate it from water. That's what the salt will be doing. Yeah, the part that has a negative charge is the phosphate oxygen groups. So the phosphate sugar backbone, the phosphates are attached to negatively charged oxygens. So that's where the charges come in. It's from the negatively the oxygens essentially that are attached to the phosphate. Okay, so we yes, precipitation means falling out of solution. Exactly, that's what precipitation is. Separation from water. Yeah, that's exactly it. Now, the other problem we have with DNA is that it is packaged with proteins. So we not only have to get it out of the nuclear envelope, neutralize the charges, we must also separate it from the packaging proteins. So that's where the proteases come in. That's where the meat tenderizer comes in. Meat tenderizers are proteases that are going to denature the proteins that package DNA. Yes, proteinases are enzymes that destroy protein. Yes, correct. Yes, proteinase. Proteases, proteinases, same thing. These are proteins. These are enzymes that destroy proteins. Correct. Okay, so we keep the DNA cold so that it remains insoluble in alcohol. The meat tenderizer are the proteinases that are going to break down the proteins that package DNA. We need a little bit of time because the enzymes do need time to work. And then in order to separate the DNA from the cold alcohol, we add the alcohol slowly. Oh, I have different slides than you guys. Okay, I would have to look at what I have. These slides may have been modified a little bit. I honestly, I would not know. I would have to look it up. But if that's the case, I'll look at what you have. Oh, you have the same ones. Oh, never mind. Okay. So there are several slides. I'm using the one about DNA isolation. I do have another one that I'm going to use next. Yeah. So the alcohol is going to sit on top of the DNA, and we're going to have these water DNA alcohol layer. Okay, so alcohol is added slowly to precipitate the DNA. Again, we have to use cold alcohol because the alcohol DNA is not soluble in cold alcohol. Yeah, and we haven't gotten rid of all the smaller molecules. We haven't gotten rid of the amino acids, of the broken down proteins and other components, but that's okay because those are very small proteins, whereas DNA is huge and it's going to come out of solution. That's when you put the rod in, it will would precipitate around the rod. Okay. So we'll go over, what is it about not cold alcohol? It's different. So I guess you would say warm alcohol. The density has changed for one thing of the alcohol. It's less dense when it's not cold. Molecules are moving faster, and they can position themselves in between DNA, and it can dissolve it. And when you say dissolve, what that means is that the molecules, and keep in mind, alcohol is a very small molecule compared to DNA. It's like water. So that means that the alcohol can position itself within the DNA. It can make bonds with the DNA. When the alcohol is cold, it's not going to make bonds as easily. The molecules are not moving as fast, and so there isn't any. It's not dissolving the DNA. For something to be dissolved, it would have to make bonds, even if it's just weak ionic bonds, with the molecule that is dissolving. That's how water dissolves. Water makes ionic bonds with whatever is dissolving. If the molecule is more dense, is condensing itself, and it doesn't have as much energy, it's not going to dissolve the DNA. So that's basically the idea behind it. So let me go to the actual lab report and go over the questions, a little bit about it to make sure that we covered that. This is it. All right, so biotechnology, the use of microorganisms to solve human problems. We did that in chapter 1. Information to synthesize proteins is encoded in DNA. Yeah, this is essentially going from DNA to messenger RNA to proteins. That's essentially the way information travels in biology, in biological systems. You should know what DNA is found in eukaryotic versus prokaryotic cells. Subunits, you should go back to the subunits of DNA, which we looked at in chapter 7. Obviously, go back to how they base pair, thymine base pairs with, cytosine base pairs with. We should know that from chapter 7. DNA is found in nucleus. No, DNA, RNA, protein is fine. Let's see, what did you say? I kind of got it. Give me a second. DNA was no. It's not tRNA. It's protein. So DNA, RNA, protein, or polypeptide. I don't think I said tRNA. I said, wait, it's DNA, RNA, protein. You can say messenger RNA. That's fine. That's even more specific. So DNA, messenger RNA, and a polypeptide. That's even more specific. But if you say DNA, RNA, protein, that's fine too. As a matter of fact, typically that's what you say. If you want to be really specific, DNA, messenger RNA, polypeptide. That's as specific as you could make it. DNA is found in the nucleus. Okay, yeah, we got that. Give me a second. Molecules codes the order of amino acids, the genetic code. Okay. Okay, so yeah, this one is one that students sometimes get confused. Let's see, the nitrogenous bases in RNA molecules are, well, you should name the 4 nitrogenous bases found in RNA molecules. And then down here, put the nitrogenous base in RNA that replaces thymine. So blank replaces thymine in RNA. So thymine is the equivalent of blank in RNA. Actually, I should say blank is the equivalent of thymine in RNA. So thymine is DNA only. The one that the nucleotide you have to put in here is the one that replaces thymine in RNA. Transcribe, you should be able to transcribe by now. Pay attention to the 5' 3' orientation. No, it shouldn't be. Thymine should not be one of the answers, because the question here is the nitrogenous bases in RNA molecules are RNA doesn't have thymine. That should definitely not be one of the answers. Remember chapter 7. Yeah. So just go back to chapter 7. The process of translation. So now you need to translate. Use a genetic code to translate the following codons. Okay, so just use the genetic code and translate the codons. Changes the nucleotide to okay. So you should know what DNA isolation is. Separating DNA from the cell, separating it from all the other components of the cell. Reagents used in the process of DNA isolation experiment. So I think we use what the sodium chloride, alcohol, water. If you don't put water, I would probably overlook that if you didn't put water. But technically, you should really put water, especially the water, whatever type of water is being used. What else was it used? The meat tenderizer or the proteases. Enzyme used to break up the cells. Okay, you should know which one was the enzyme that was used to break up the extra of DNA. So go back to question 2, I should say, sorry, and figure out what is the enzyme. The ethanol is added slowly so that we can precipitate the DNA, so it doesn't go back in solution. And DNA is collected with that little loop thing. What does it look like? Describe what it looks like. And the exercise tells you this is what it looks like if it's broken, and this is what it looks like if it's not broken. Okay, so you give me a picture of the gel. Am I in the same place? Did I miss something? 6. There we go. Okay, never mind, that's better. All right, so now we're going into gel electrophoresis. Okay, so let me stop here. Any questions about DNA isolation? It should be fairly straightforward. It should not be too bad a lab, actually. Now, let me go for DNA isolation. Oh, sorry, not isolation, but the gel. Okay. I do have another. Yes, and I know. You can do it at home. Yeah. You can do it at home, actually. You can get strawberries at home, get all the reagents. You could do this at home. This is one of the experiments that you could, one of these days, I may even actually do it so that students do it at home. Yeah, you can do it at home. Alcohol, meat tenderizer. Oh, I see. Well, okay, that's a different kind of problem here. Let's see, give me just a DNA isolation. That's with that already. DNA technology, that's the one, I think. It's just a second. I just downloaded everything. Yeah, I'm pretty sure this is it. Okay. All right. So this is the idea behind it. If you put DNA in an agar gel, so essentially you make a block of agar into the form of, you take agar and you let it solidify in the form of a gel, a block, I should take. Just a second, trying to drop the block. Okay. Now, it may look solid or semi-solid to us. It is not really. At the molecular level, that little agar gel is actually a sieve that has holes in it. Obviously, we don't see it, but it does. Okay, so that we can use it to separate DNA molecules, because if you put the DNA molecule on one end of the gel, and then you put the gel in a buffer, and you put current going through the buffer, but you have positioned the DNA so that it is in the negative side of the electrophoresis chamber, and the positive side is on the other side of the gel. DNA being negatively charged, when you put electricity through the gel, is going to naturally begin to move towards the positive end, because opposite charges attract. Now, because this is a sieve, the larger the molecule it is, the longer it takes it to weave its way through the sieve. The small DNA molecules, because they are small, are going to very quickly travel through the little holes and reach the positive side. So that's how the gel separates DNA according to the size of the fragments. The smaller fragments travel quicker through the gel than the larger fragments, because they are big and they're snaking around in there, and they take them longer. Okay, so that is the idea of gel electrophoresis. Gel electrophoresis can be used for, identify paternity, DNA studies, diagnostics, infection agents. There's all kinds of uses for it. But obviously, what you would need to do with the DNA first is cut it into fragments. And in order to cut DNA into fragments, we use special enzymes that cut DNA, called restriction enzymes, that we actually get from bacteria. And restriction enzymes will cut each DNA differently, creating different size fragments. Okay. Okay, so you want to establish the genotype of individuals with regard to a gene that produces the protein TNF-alpha. Okay. All right, so essentially they're working with a gene, and the gene is going to have A's and T's and G's and a pattern of these nucleotide sequences. I'm just making these up right now here. So if we say, okay, this is the normal pattern, but some people have mutations. Okay, let's make a G instead of a C, and then C, G, G. Okay, so there's a mutation right here. This is a mutation right here. Okay, so you use restriction enzymes that are going to cut this gene into pieces. In a normal gene, okay, does the term stablish means the same as, oops, never. Yes, it does. That's a misspelling. I don't know why it was not cut. Yeah, that's established. Yes, it does. It's the same as established. This is, I don't know why it didn't catch it. Yeah, to establish the genotype. Yeah, to determine the genotype of individuals. Yes, thank you. Yeah. Yes, yeah, it is. That's me. Mm-hmm. And it doesn't give me the little line underneath telling me there is sometimes I don't catch these misspellings. So it allows us to determine the genotype. Genotype has to do with the sequence of nucleotides in the DNA versus phenotype. Okay, so produces, but yeah. Okay, so we want to know whether there is a mutation in a gene of or a protein called TNF-alpha, tumor necrosis factor alpha. Okay. So it's a protein that immune system cells make to eliminate abnormal cells. And in some people, that protein is mutated, so they cannot make the proper protein, and so they're more prone to having tumors because the immune system doesn't make the protein that helps these cells, these immune system cells, recognize abnormal cells. So if you have a normal protein, when you use restriction enzymes, that this restriction enzyme, let's say, And by the way, restriction enzymes cut DNA in very specific patterns because restriction enzymes recognize DNA sequences. So let's say that they use restriction enzyme X that recognizes the sequence ATC, and when it sees that C is going to cut right there. So that if you have a normal gene, you will end up with two fragments, one with 3 nucleotides and the other one, I guess, let's make another nucleotide here, with 4 nucleotides. So one would be a little bit large fragment than the other. On the other hand, the mutated gene doesn't have a sequence that the enzyme can recognize. So let's say it either won't be cut or it will be cut in a different manner, so that it will have a different pattern altogether. Okay. And so that's essentially how they're going to detect which patient has a mutated gene, is by comparing the pattern of cuts. Is there a restriction enzyme for every gene code or nucleotide? No, but there are restriction enzymes. We have a whole arsenal of restriction enzymes that recognize certain nucleotide patterns. And this is the deal. The example I've given you, I only have, 10 nucleotides. In a gene, you're going to have 1,000 nucleotides. So you will have restriction enzymes that will cut 1,000 nucleotides in a specific pattern. So no, there isn't a restriction enzyme for every gene code, but we have enough restriction enzymes out there where you will, and you'll typically use more than one. You'll use 2 or 3 restriction enzymes, and at least one of them is going to find a place to cut within a gene. Typically, more than one of them will find a place to cut in a gene. Okay, so even if, let's say, you use 3 restriction enzymes and none of them cut the gene, then you go back and use 3 more. So and by the way, restriction enzymes are harvested from bacteria. We find them in bacteria. And when we found them, we began using them in research. And all bacteria produce restriction enzymes. And that's what I'm saying. We have a whole arsenal of restriction enzymes that a researcher can buy and use it. Yeah, there's just example. That's an example of biotechnology. Exactly. Yeah. Bacteria uses restriction enzymes to protect themselves from viruses. So those are the enzymes they're going to use when they're being infected with a virus, trying to destroy the viral DNA. So yeah, there are tons of them, and they're very, very useful in research. And that's one of the uses, is to cut DNA in specific patterns, and we know, okay, this is the normal pattern. Now let's find whether this patient has a normal gene or not by the pattern that we get with them. So, and in this case, they're giving you a pattern for the normal gene, and they're giving you the pattern for the mutated gene. So it's just a matter of running the gel with the normal and the mutated gene, and then comparing it to the patient. So this is the deal. That's just showing you what an electrophoresis gel is like, how to put it together. Those are the different parts. This is the tank where the buffer is going to be put. This is the little tray where the gel is going to be poured. It typically has some type of piece that is put at either end so that the gel is liquid, so it doesn't spill, because you pour it as a liquid and you have to wait for it to solidify. Let me see. Yeah, there you go. So you put little devices here that is actually, it can be something as simple as tape. You can put tape right here and right here to make this into a box. And you have the little comb in there. So you put the liquid gel on the tray and wait a few minutes for the agar to cool and solidify. Then when you take out the comb, now you're going to have little wells in the cast gel. So you're going to have little holes in there. That's where you're going to put the DNA. Okay. So you take once the gel has solidified, you can take the tray with the gel. Okay, there's pouring the gel, and this is actually just tape that they have put at the ends to make the box. Let's see if it has them. Okay, maybe not. I'll come back to this in just a second. Then you put the tray down here in the chamber with the gel. You put the buffer. This is already set up, electrodes, and obviously this is the top. This will be the negative electrode. That's the positive electrode, and you can connect it to a power source and begin running the electricity after you load the DNA. Okay, so you remove the tape. You put the tray in the electrophoresis chamber. The buffer is there to conduct the electricity. Both the gel and the buffer has a dye that allows us to see the DNA. Ethidium bromide, which, by the way, if you go back to chapter 8, that's a mutagen, is often used. Binds to DNA. It's an intercalating agent. That's what ethidium bromide is. And it is a fluorescent molecule. So it's going to intercalate into the DNA. And when you put it under UV light, the DNA is going to fluoresce and you can see the bands. Okay. Let's see. Let's go for the sample. Yeah, the other dye that's used is a bronthymol blue, and that allows you to see the DNA when it's being migrated. And I'll show you that in just a second. Okay. All right, so let's just draw a little gel here. And you have the wells. The DNA is put in the wells. There's also a loading dye that is mixed with the DNA. The loading dye is going to do two things. That's the bronthymol blue. It's going to do two things. It's going to dye the DNA, but it's also going to weigh it down so that when you put it, you load the wells when the DNA is in the chamber and the liquid buffer is there already. So you're going to go through the buffer, through the liquid, and load the DNA into the well, which means that unless the DNA has something to weigh it down, it will come out of the well and begin to float. So that's another purpose of the loading dye, is to weigh the DNA down so that it stays down. So let's say that the normal pieces of the gene have two fragments, and one small fragment and one large fragment. So once you set up the gel, the small fragment will travel quicker, the big fragment will take a little bit longer. So that's how they will be separated. Let's say that the mutated gene has, let's say, one small fragment and one medium fragment, and let's say one large fragment. So that one will have maybe a little fragment here, a fragment here, and a fragment here. That tells you there's 3 fragments, not 2. So it's definitely not the it's definitely the mutated one. So you compare that to the patient, and if the patient ends up with the same fragments as the mutated one, then that means the patient has a mutated gene. Okay. The other thing that they do is they put what they call a ladder. They load a DNA fragments of known size. The ladder is something that is purchased. So you buy the ladder. It already comes with the DNA cut up, and the fragments are known, so that you end up with these series of DNA fragments. And when you look up the sizes, it's going to tell you, okay, the top one here is going to be, I don't know, 1,000 base pairs. And this one would be, I don't know, let's say 800 base pairs. The one down here would be 50 base pairs, so that you can, you know, the size of each one of these fragments. You can approximate the size of your fragments by comparing it to the ladders. You can say, okay, this top one is about the same place as the top fragment on the ladder, so it's about 800 base pairs. This one down here will be a small one, about 50 base pairs. So you can have an approximation of the sizes. Actually, there's more than an approximation. You can actually measure the distance of each one of these fragments in centimeters, and have a little standard curve of base pairs versus centimeters, and then measure the distance on your fragment per curve and say, okay, if I have this many centimeters, that means I have this many base pairs. So that's even more specific, more accurate than just comparing. But for our purposes, we just compare. So that this first exercise is just to figure out whether the patient has or doesn't have the mutated gene or the normal gene. So it says, which fragment will separate further from the well? Well, the answer is, the smaller the fragment, the further it travels in the well. Okay. So the bronchial blue allows you to see the DNA without UV light. So as it is traveling through the gel, you have an idea. It's a little bit hard to see, but at least you have some idea of where the DNA is, so that you don't let it run for too long. If you let it run for too long, the DNA will run the entire length of the gel, and you'll lose it in the buffer. Okay, so that's the first exercise. Let me go and see what the questions are. Yeah, typically you start with about a 30-minute run, 15, 20, 30 minutes, and then you stop and see where you're at, and then decide whether you want to run it more or not. But that's about how long you typically run it. About 30 minutes. If 30 minutes, again, I would stop at 15 minutes, look at it, see what's going on. That's not enough. Let's run it another 5 minutes. Let's run another 5 minutes. At 30 minutes, should be about the same. Okay, there you go. So the lab said to do it until the bands migrate about 70% of the length. Correct. So most of the length. If you let it go too long, you're going to lose bands. Yeah, you're going to lose the smaller bands. So that's why you stop and you keep looking. The problem with not until the bands are separated. Yes, that's why you stop and look to make sure the bands are separated. However, keep in mind, you want to make sure you catch all the bands. If you don't wait long enough, the bigger bands may still be in the well. So let's say you stop at 15 minutes and what you see is 2 bands, but there's still a big band in the well that hasn't completely, let's say that big band is somewhere in here, it's not quite separated yet. You run it 5 more minutes and all of a sudden you have 3 bands because the bigger band may not have separated yet if you don't run it long enough. So that's the only thing about not running long enough, is that the bigger bands may still be trying to weave their way out of the well. And you may miss the big ones. Little ones will come out very quickly. Okay. Yeah, this exercise was pretty straightforward. Correct. Yeah, this exercise was straightforward. There were only 2 bands. And for most, I think, if I remember correctly, pretty much everybody gets this one. It's not very picky. So I'm going to go back to the lab report and go over the this particular part of the lab report. You should have been okay. Correct. You should have been okay without having to wait for 70% of the bands, for the bands to travel 7%. You should have been okay. You should have still gotten the correct answer. Okay, you should know which side to load. Keep in mind, opposite charges attract, so you want to load on the same size charge so that the DNA migrates to the opposite size charge. Just keep in mind DNA is negatively charged. Yeah, we got that one already. What part of the molecule gives the charge? It is applied. How long did it take to separate the DNA fragment? Okay, so just put down how long you waited. How many DNA fragments were loaded in the gel, including the control. Okay. All right, so you should come up with the two controls. You have two controls? Yeah, you had two controls plus the patient. Okay, a second. Okay, there we go. That it? Yeah, that's it. Okay. That's not it. All right. Okay, provide a picture of the gel. I guess not. It's a very simple lab. I couldn't come up with any extra credit. Provide the picture of the lab notes chart or use the table provided instead of allowing. Yeah, this is one that everybody gets good grades on. Okay, you got that. Let me see something. NF2. Okay, so you have two patterns to compare to, and if the patient is homozygous, meaning is healthy, has the homozygous for the normal gene, then the pattern will match those of the normal gene. If the patient has only mutated genes, they will match the mutated genes. Keep in mind, in humans we have two copies of a gene. So if a patient has one good copy and one bad copy, you're going to end up with fragments that match both patterns, the normal and the mutated one. So which one travels the furthest, TNF1 or TNF2? Again, you have to look and decide that. Which family is the largest? Again, the one that travels the furthest is the smallest. The one that is closest to the well is going to be the biggest. So this is a fairly easy lab. Lab report 9 and lab report 10, especially lab report 9, we're going to go back to gels. But now we're going to use a ladder, and we're going to have more fragments, and we're going to do 2 gels for 2 different purposes. So this is a good practice, good introduction to this type of exercises. Okay. So this should not be too bad. Any questions? Okay, so we'll meet again on Thursday. Oh, yeah, you have a question. Go ahead. Yes, what part of the molecule gives the charge? Yes, I'm asking about, no, I'm asking about the DNA. So what part of the DNA molecule is given the charge? Is the one that is charged. Because what do you have in a DNA molecule? You have the sugar, you have the pentose sugar. So go back to chapter 7 and take a look at the DNA molecule and see if you see any charges. So that's the sugar. The bases are again just rings of sugar with nitrogens in them. Then you have the carbon outside. You have the phosphate with oxygen. Oxygen is the one that's going to end up with charges. Okay, oxygen is a charged molecule, especially in a water environment. In a water environment, oxygen will be charged, just like in water.